Hello and welcome to my channel. This is your tutor doctor from DB Tuitions. This is part 6 of GCSE AQA Biology, paper 1, higher for triple science. And in this video, we're going to talk about question 6 of your past paper from June 2024. This is a walkthrough where I'm going to solve the question 3 and give you detailed explanation. Now, this question is all about pathogens, antibiotic resistance, and viruses. And I want you to pay close attention because this is one of those questions where students think they understand it, but examiners feel otherwise. We're going to go part by part, decode the petri dish diagram, look at why resistance is misunderstood, and I'll show you exact phrases that are going to score marks. If you have not followed my channel, I strongly recommend do subscribe to it. The name of the channel is DB Tuitions 09, both on TikTok and YouTube, and DB Tuition 09 on Instagram as well, so that you're not missing out on any important videos before the exam. Now, let's look at the first question, 6.1. As I always recommend, first, you go to eyeball the marks. This 6.1 is 1 marker. Let's read about the data given for this question. This question is about pathogens. A scientist investigated antibiotic resistance in bacteria. Now, I want to do a quick explanation. Pathogens are disease-causing organisms. Pathogens release antigens. A quick lesson here. Pathogens end in gen, antigens also end in gen, they match, therefore pathogens release antigens. Now, both pathogens and antigens are being fought. So our body is going to fight the pathogens. It is going to kill the toxins, the antigens released by the pathogens, and that's called antibodies. So antibodies fight antigens, they also fight pathogens, and antibody is released by our body. So please do remember, our body produces antibodies, and pathogens, which can be a bacteria which is causing an infection, a virus which is causing an infection, so it ends in gen. So pathogen releases antigens, and our body releases antibodies which fights with the antigens and pathogens. Now, having explained that, question 6.1 says name one antibiotic. So I'm going to give you a few antibiotics, but this is only a 1 marker. You are required to only name one of them. The most common one is penicillin. Now, amoxicillin is also having penicillin, so it is the same category of antibiotic. Then we also have tetracycline. I'm giving you the 4 most common ones, and then we have erythromycin. So if you learn these 4 for your exam, it's more than enough. It is very important that spellings are important in this case. If you get your spellings wrong, they're going to mark you down. Remember, 1 correct answer is better than giving 2 answers in which 1 is correct and 1 is wrong. If you do that, you will end up not getting the mark. So please only put 1 answer for 1 mark and make sure you put it right. This is a simple recall question. You just need to name 1 antibiotic. In this case, penicillin should be more than enough. But for your revision, I have given you 4 of them. Please do not name any vaccines. Do not name Nurofen, ibuprofen, paracetamol. These are painkillers. We are looking antibiotic. Anti is opposite, biotic as in bacteria, or not virus, sorry. Only antibiotics are the ones which kill the bacteria. Now let's move on to question 6.2. Now let's look at the marks. It says 2 marks and there's a figure given to us, and we're going to read the information given along with that figure. The scientist grew one type of bacterium on agar in a petri dish. The scientist placed paper discs, each containing a different antibiotic on the agar. The figure 5 is showing the appearance of petri dish after 2 days. In figure 5, we can clearly see the agar plate. The gray area is the area where the bacteria is going, and there are 3 discs, paper discs, on which we have 3 different kinds of antibiotics, and they've just named the antibiotics as antibiotic A, antibiotic B, and antibiotic C. And you can see that around antibiotic B, we have this white zone, which is known as the clear zone. Okay, both for antibiotic B and C we have that, but around antibiotic A we don't have any clear zone at all. Now the key skill for this question is interpreting a petri dish. What is clear zone? The white area here, the clear zone, is actually telling us that the bacteria in that white area around the black dot for both antibiotic B and C means the bacteria is killed. So the antibiotic B and antibiotic C are able to kill the bacteria. And in this case, you can see there is no clear zone around antibiotic A. It was not successful in killing the bacteria. So this bacteria is able to dodge the antibiotic A, but it was not able to dodge antibiotic B or C, more so B than C. You can see that there is a larger clear zone around B than C. Now that we have understood the petri dish, we are just going to go ahead and answer the question. And before doing so, around A, it is a no clear zone. So these are the phrases you're going to use in the exam. Clear zone around B and C antibiotics, and a no clear zone around antibiotic A, which mean antibiotic A, so the bacteria has resistance for antibiotic A. Now let's go ahead and answer the question. So 6.2 says, students said the bacterium is resistant to antibiotic C. Explain how the results in figure 5 show that the student is not correct. So they're already telling us that the student is not correct. So we are going to say the bacterium is not resistant to antibiotic C, right? Why? Because there is a clear zone around antibiotic C. So we're going to write that, and what we're going to say, it is showing that bacteria has been killed or have been killed. I'm just writing it, and cannot grow near it. So that is showing us that antibiotic C was able to kill the bacteria around it, so therefore it has a clear zone and bacteria could not multiply again. And therefore, this is how we're going to answer this 2 marker. Now, always remember, clear zone is killed. No clear zone around the antibiotic paper disc. It means that bacteria is resistant to that antibiotic. Now, in this kind of questions, what are the common student errors? I've seen as an examiner some students writing, it works better. No, that's not the language you want to use in an academic GCSE paper, or saying less bacteria. No, it's a clear zone. It means there's no bacteria growing in the clear zone. The important phrases, takeaway phrases for this question is clear zone and mentioning that that antibiotic has killed the bacteria. Those are the important mark scoring phrases. Now let's move on to question 6.3. Again, this is a 2 marker as we observe. Now it says, suggest why doctors are concerned about antibiotic resistance. Now, the examiner is looking for your ability. So all my fams, pay attention here. Are you able to establish a cause plus consequence? This is what they are looking to see here. Now, we are going to say doctors are concerned about antibiotic resistance because some bacteria are no longer killed by antibiotics. Yes, in hospitals, there are some very deeper infections which are not able to killed by many of the antibiotics. This means infections become the next point scoring statement or phrase. The infections are difficult to treat and therefore spread more easily, because if we are unable to treat those infections are able to spread and can cause serious illness or death. Now I'm going to write a mnemonic here called risk. So in risk, R is resistant bacteria survives. Remember that. Resistant bacteria survives. Infections become harder to treat. So the infection is harder to treat. I'm just repeating so that if you're noting it down, you can note that down as well. S, the spread. The spread of the infection increases. And K, it can kill the patient. So you must mention treatment failure. Vague answers will not score more than 1 mark. So let's be precise for this. Very well done. If you've already got this right, let's move on to question 6.4. Now, diseases caused by viruses cannot be treated using antibiotics. Now, suggest why viruses cannot be grown on agar. And this is just 1 mark. They are just looking for you to identify the difference between bacteria and virus. Remember, bacterial cells are unicellular mostly and prokaryotes, whereas viruses, they need living cells. So all of you remember recently we had a pandemic, COVID, not great times, but COVID-19 was a virus. It can only take over a host cell and it can hijack the nucleus and have its DNA in place of the cell's DNA and start multiplying. So viruses cannot be grown on agar because they can only reproduce. They can only reproduce inside living cells. So it's very important, because agar does not have any living cells, viruses cannot be on agar. They cannot grow on agar plate. Now, common mistakes what my fams make here, they kind of, read sentences as an examiner. They need food. They are too small. Come on, fams. That's not the reason. Living cells is the only accepted idea for this kind of question. Now let's move on to question 6.5. It says, why is it difficult for scientists to develop drugs to destroy viruses? Again, this is a 1-marker question. Now, the key concept behind this is viruses use host cells. So you need to develop this idea so that you can gain marks here. It is difficult to develop drugs to destroy viruses because viruses live and reproduce inside body cells. So drugs that kill viruses they can also damage body's own cells. Because remember, if they are testing out a drug, let's say we are testing out a medicine, which we have made to kill a virus, but the virus is growing inside the body's own cells. So we end up targeting the body's own cells as well, and we can damage. So the correct answer, as viruses uses host cells, the drug may also damage body's own cell is the right answer. Now, kill virus, you risk killing the host cell. That's your memory phrase. Very well done. If you've got this right as well, let's move on to the last one. In question 6, which disease is caused by a virus that damages white blood cells? And again, this is a 1 marker. Now, this is an MCQ. Now, the correct answer here is AIDS, which is caused by HIV. HIV is the virus which attacks white blood cells and it weakens the immune system. Whenever you're answering MCQ style of questions, quick elimination is a good tip to follow. Now, gonorrhea is caused by a bacteria. Salmonella is also a bacteria. Now, remember, measles is a virus. Measles is a disease caused by virus, but it does not attack white blood cells. So it's important to know the names of these diseases. So gonorrhea caused by bacteria. Salmonella is again a kind of bacteria. And again, you have measles, which is caused by virus, but it does not attack the white blood cells. Now, once again, giving you a quick summary of question 6. Remember, clear zone means antibiotic is working. No clear zone means resistance. Drugs which are antibiotics do not work on viruses, and viruses need living cells. They take over the nucleus of a living cell to reproduce. And AIDS is a disease which damages white blood cells. And that completes question 6, a very important topic linking biology to real-world medicine. If you can confidently explain antibiotic resistance and viruses like this, you're scoring and securing marks that many students miss. If this video has helped you, make sure you subscribe to DB Tuitions, because this is part of our full AQA GCSE Biology Paper 1 question by question series. It is built to help students and also parents understand exactly how examiners are awarding marks. And I'll see you in the next video. Bye-bye.